Hello everyone, welcome back to Dr. K. PREM-PRIMER Biotech Lectures. Today's topic is features of cloning and expression vector. Before cloning of your gene, when you are planning to clone any of your genes, right, you should know the features of that vector. Once we know the features of that vector, we will be knowing that whether that vector going to be useful for your purpose or not. Hence, I am trying to summarize both the cloning and expression vector features. Probably this lecture may help you to, in selection of the vector and their features, right? As you all know that I am Dr. K. Prem Sagar, the Department of Biotechnology, Mahatma Gandhi University, Nalgonda. And don't forget to subscribe my channel. That's Dr. K. Primer Biotech Lectures for many updates. Here I will list out the various features of vectors, both cloning and expression vector, one by one. And also I'll try to explain the importance of that vector, feature of that vector. The first one and foremost one is the vector must have its own origin of replication. When a vector have its own origin of replication, then replication of that vector will be autonomous or self. So as you all know that replication begins at the origin of replication. That's a site or a nucleotide sequence where the DNA molecules are get unwinded and initiate the replication. So vector means it should be an autonomous replication vector. So origin of replication is the most important element to be there in the vectors. The second one is that once a vector have origin of replication, the second most important is that multiple cloning site or polylinker. So multiple cloning site is nothing but a bunch of restriction sites which are there, which will allow the cloning of your gene of interest in unidirectional or bidirectional. MCCS, that is multiple cloning site or polylinker, allows you to clone your gene of interest in a uni or bidirectional clone. Uni means you can use the single restriction endonuclease site, or bidirectional means you can use the two restriction sites to clone so that you have a control over the orientation of the DNA fragment. Third most important one is genetic selection marker. When you clone your gene of interest into the MCCS, that's a multiple cloning site, and then you transform into the system, maybe E. coli, yeast, or mammalian cells, then you need to select the transformants. Transformants could be with an empty vector or recombinants. So when you should have a transformants, transformants can be selected when your vector have a antibiotic resistant gene. Antibiotic resistant genes. Naturally, they act as a genetic selection marker. So where vector have a antibiotic selection marker, you cloned, and that vector is transformed into the E. coli, for example, then that E. coli can grow in the presence of antibiotic, right? So non-transformed E. coli or other systems will die. Only transforms are survived because of the presence of genetic selection marker in the vector. And also there is a need to have a second genetic selection marker for the selection of recombinants. You see, one genetic selection marker for the selection of transformants from the non-transformed, which will die, and the second genetic selection marker to select the recombinants. So here, the MCCS is there in the second genetic selection marker, and when you clone your gene of interest, then the second genetic selection marker will be disturbed, and only vector will have a second genetic selection marker completely, and recombinants without or disturbed genetic selection marker, so that transformants and the recombinants can be selected. Especially when you want to clone your gene of interest, to get the more copies of it, that vector must be high copy number. High copy number. So when you clone your gene of interest into the high copy vector, so that the each cell have many copies of that vector, and there is a high chances to get your copy of gene in large number or large quantity. And the sixth point and sixth feature is small size of the vector. When the vector is small sized, so that you can increase or you can clone the larger DNA fragments. Larger DNA fragments can be cloned when vector size is small, so small, right? So, if you take an example, PUC18, it is around 2.5. So you can clone another 7.5 kb of DNA fragment, right? So these are the six general features which are present in the cloning vector. And I'm going to talk about the another 7 features uh, with the 6 and another 7 features are present in the expression vector. You see, the 7th one is strong and inducible promoter. When you clone your gene of interest into the expression vector, downstream to the promoter, downstream to the promoter. So that promoter must be strong and inducible. Strong and inducible means strong promoter initiates the transcription at high level. Transcription is very high. More copies of cloned gene is transcribed and you get more copies. The second most important one is inducible. So when you clone your gene of interest under the control of strong promoter, it must be inducible. Means whenever you want to initiate the transcription by adding a inducer, that time only transcription has to be started. That time only transcription has to be started. That's a reason all the expression vectors have both these two features, strong as well as the inducibility. Strong and inducibility. And the 8th point, strong terminator also very, very important. Strong terminator means at the end of your gene, there is a terminator sequence. That terminator sequence will stop the transcription of your clone gene so that there is no further transcription. So strong terminator. And the 9th point is that ribosome binding site. You all know that ribosome binding sites in case of prokaryotes, Shane-Dalton sequence, in case of eukaryote Kojak sequence, right? That sequence must be there after the promoter or at the end terminal of your gene, right? So you will be cloning only ORF. ORF means what? This sequence starts with the ATG, that start codon to stop codon. May not have its own ribosome binding sites. Hence, the vector should also have a ribosome binding site so that it initiates the translation. And the 10th point is that protein tags. Protein tags, they are very small. which are very, very helpful in the purification of the recombinant protein, detection, and the quantification. For example, there is a histag. Histag means 6 histidines or glutathione transferase. Both are there at the N terminal of the N terminal of or downstream to the promoter after ribosome binding site. These is tags and GST tags are there. After that, you will clone your gene of interest, so that your gene of interest N terminal will be contains of is tag or GST tag, so that when you express your gene of interest, right, then you get a protein. That protein with the is tag or GST tag will be there. That is tag and GST tag is useful for this single step purification. You all know that E. coli have 4,000 proteins. Out of that, your protein is having only is tag or GST tag, and you can use the complementary substrate in the affinity column, like histag, you need NTA column or glutathione, so that your recombinant proteins are purified in a single step. And also you want to quantify, right, with the Eastern blot, or so that these tags will be very useful. And I here F1 origin. This is a F1 sequence which is used to for cloning of sequencing of clone gene. When you want to know structure of a gene sequence, you have to have a F1 origin. That F1 primer can be used to sequence your gene. Secretory signal. When you are cloning your gene of interest into the expression vector, and that expressed protein should not go to the vacuoles or in the host system. If they can release into the media, so that purification would be very easy. That's a reason alpha mating factor of yeast are kept before the your ORF, and then your your protein will be having alpha mating factor. That alpha mating factor will aid in the release of or a secretion of your recombinant protein into the media, so that you'll have a easy procedure to purify the recombinant protein. That's the reason secretory signals are kept. And the localization signal, you can use GFP, green fluorescent protein, from the jellyfish. Here when you want to know subcellular localization of the any protein, right? You can clone your gene and transform into the native system with the GFP, and your protein and GFP will go to their native location, and under the blue or UV light, they'll be emitting signal, and you'll be knowing that particular protein localization. It may be a mitochondria, it may be a nucleus, it may be a Golgi complex, it may be a plasma membrane. So to locate your interested gene or their product location, if you add this GFP protein to the at the C terminal, and then you will be knowing that particular protein location in the native system. So these are the 13 different features, but all of them may not be there in the same vector, but depends on your need. You can choose the vector with these features. I'll show you different vectors with the different features in the next slide. You see, you all very much know about the 18, right? So small, you can see here, this is the origin of replication, helps in the autonomous replication. This is a multiple cloning site. To clone your gene of interest, you see a bunch of restriction sites. And this is a genetic selection marker, that's nothing but beta lactamase, helps in the selection of transferments from the non-transferments. And this is the LAG-Z prime. That's another genetic selection marker, where to help the recombinants from the non-recombinants or just transferments. And the small size, as I told you, that small size. When your vector is small size, you can clone bigger DNA fragments. In case of POC18, you can clone almost 7.5 base, 7.5 KB vector to make up to 10 KB, right? And the sixth one is uh, high copy number. So to facilitate the get the more copies of your gene, if that vector is high copy number, you'll have more chance to get the more copies of your gene. And the next one is a PET21 most popular expression vector. In the previous slide, we have seen the six features which are very important to the cloning vector. Here we are going to talk about the expression vector. In addition to the cloning vector features, other features are very, very important here. I'm going to talk about them here. That is a T7 promoter. That's a strong promoter. T7 RNA polymerase driving the T7 promoter to express your cloned gene. And the terminator, as I told, to cut there itself, the strong terminator is there. And the ribosome binding site. I told you that ribosome binding site is very, very useful, and that is required for the initiation of translation. You can see immediately after the promoter, there is a ribosome binding site, and followed by the T7 tag and histidine tag also there. You can see histidine tag in this one, right? Histidine tag and F1 origin for the sequencing of clone gene, right? So here, in addition to the features we discussed with the POC18, these are the additional features in the expression vector, and also we will take some more examples of the expression vector to see the other features. You see, this is a PET E1. You can see is tag that helps in the purification as well as detection of the recombinant protein. And here you can see GST tag in the PEX and GST. That's a glutathione S-transferase there. And this helps in the solubilization of the recombinant protein. And you can see here alpha factor. That's a secretory signal of yeast. It is there in the very N-terminal region of the cloning sites, so that it will be included in your gene, and your protein will have an alpha factor. That alpha factor guides the protein to secrete into the media. And here, as I told you, that GFP, green fluorescent protein, right? That's there in the N-terminal region of the MCCS, and then this GFP and your protein will goes to the native location and tells you the exact location of that protein location in the cell, and that is where we can decide the function of that, right? So this is all about the features of cloning and expression vectors used in the biotechnology, right? So this is all about that. Hope you like this lecture. If at all, try to subscribe my channel. That's a Dr. K Primer Biotech Lectures. You will get updates, whenever I make a video. So don't forget to do that. Thank you all for listening. See you all again until next video. Bye-bye.